All this is Dr. Mobeen Sayed from drbeen.com. Welcome to one more lecture. Today is the second lecture for the function of the immune system. We are looking at the overview of the immune system. What I would love to take away from today is number one, to look at the remaining cells of the innate immune system, that is granulocytes, eosinophils, basophils, neutrophils, mast cells, um, and then we will also look at NK cells. That is one. Secondly, I would like to become aware, we together, of the process of phagocytosis at a high level, and then also understand the definition of antigen, because phagocytosis is a term that will come up again and again. Antigen is a term that would come up again and again. And these cells, we would discuss them repeatedly. So this still is a high level discussion. We would go in depth in the future lectures. Let's start our discussion. And I am using OBS today. So if there is any issue, please let me know. So let's start our discussion. So the effector cells of the innate arm. So total cells for the innate arm, we did some yesterday: macrophages, dendritic cells, monocytes that give rise to macrophage or dendritic cells, then granulocytes, which are neutrophils, eosinophils, basophils, mast cells as well, and then natural killer cells. There are other cells too, but these are the more important ones. So today we'll do the remaining cells. So what are these cells? If we just very quickly look here, this one is a neutrophil. We'll discuss more, but just know that it stains light pink when it is stained with right stain. And neutrophils have a very interesting thing. Look, I made this little burner in them. Keep that in mind for the whole immunology. If you can just remember one thing about neutrophils, and that is respiratory burst or respiration, or this little burner inside the neutrophil, that is sufficient to at least know what is the function of the cell. Then is the eosinophils. Eosinophils are usually red in color. We'll discuss later, but just eosinophils, they appear red. That is why they're called eosinophils. Basophils and mast cells, they appear blue. That is why they're called basophils or basophilic system. Eosinophils are also called acidophilic cells. These three cells together are called granulocytes because when you see them after staining under a microscope, they look like they have a lots of granules in them. They have a lot of tiny dots in them. Today we'll talk about the NK cells as well. If you see the NK cells under a microscope, you might actually see them wearing an eye patch and holding these little things. So we'll discuss them as well. I'm kidding, of course. So first concept, innate arm primary function, or the way it attacks the pathogens, is by recognizing patterns on them. So if you look at these, this is a virus here, this is a fungus, this is a bacteria. Maybe I should have made a protozoa over here as well. But if you look at these microbes from the eyes of the innate arm cells, especially phagocytic cells, which we would discuss today, but these are macrophages and dendritic cells, neutrophils as well. If you see these cells, these microbes from these cells' point of view, you would see patterns on them or patterns on them. So somebody was trying to correct my pronunciation for this one yesterday. So thanks to them, I'm going to try to say patterns instead of patrons. So we'll see how successful I am. So here, there are patterns on these microbes. These patterns are something that our innate cells can look at. And not only they can identify them, they have pattern recognition receptors, but once they can recognize a pattern and identify that this is a microbe, then these cells can phagocytose them. They can eat up these pathogens and break them down. So that is the process that we're going to look at now. So here, imagine there is this little bacteria standing here, and this bacterium is standing there doing nothing, but a macrophage that is standing nearby is seeing patterns on this bacteria or bacterium, the cell. And macrophage has become very interested. Macrophages actually have these pattern recognition receptors or PRRs, and these are the pathogen associated molecular patterns, PAMs or DAMs. So here you see a macrophage becoming interested in this bacterium. Then what it would do is this macrophage will walk to this bacterium and start touching it and sensing it. Of course, now it is sensing the patterns on this bacterium. Once the macrophage finds out that there is a pattern, this is a microbe, then what macrophage does is it starts creating a pocket around this bacterium so that it can engulf it, so that the macrophage can engulf this bacterium and bring it in. This process, as you see here, these little extensions are now going around this bacterium, and slowly they would completely surround it, and this bacterium would become a small vesicle or a pocket that would be incorporated in the macrophage. This happens with the dendritic cells as well, and this happens with the neutrophils too. This process is called phagocytosis. Phagocytosis means eating by the cell. Pinocytosis means drinking by the cell. When we pick up, or the cells pick up these large structures, that is the bacterium or viruses or fungi, this is called phagocyte. Even debris, a macrophage can actually eat up another macrophage. Normally, a macrophage can do 100 to 120 phagocytosis before it dies. And in that process, macrophage doesn't have to just pick up a bacterium. It can pick up a dead cell, it can pick up debris, it can pick up bacterium, and so on. Okay, so back to the phagocytosis. Now the macrophage is surrounding this bacterium. Then, as you can see here, it has actually built a wall around this and brought it inside its own body. So now here, this bacterium is trapped in something that is called phagosome. And this phagosome contains the bacterium. When it is inside the macrophage or neutrophil or dendritic cells, they all do phagocytosis. Then what happens is, neutrophil has a different function. They do the respiratory burst. We'll talk about that. Macrophage would connect digestive enzyme vacuoles. These are called lysosomal vacuoles. These little vacuoles here. These are connected with the phagosome, and the digestive enzymes are spilled in the phagosome where there is this little bacterium or microbe. And those digestive enzymes break down the microbe into tiny, small pieces. For our example, you can think of a glass of water, a glass made up of actually glass. And if you break it, it will break in a lot of tiny pieces. That is what would happen to this bacterium or this microbe. Now, here is a very important and a delicate point. That point is, so see here that this microbe has now been broken down. When the microbe is broken down into smaller pieces, some pieces are going to be very small, some pieces are going to be relatively big. However, some pieces would be just the right size for what? The right size that B cells of the adaptive arm or T cells of the adaptive arm can bind with it. With what? With that little piece. So, what does that mean? If you break a glass, you would see the pieces are of different sizes. Not every piece can simply be called an antigen. Technically, these are all antigens. But by definition, an antigen is a fragment of a microbe that can be bound to a T cell receptor or a B cell receptor. This is a very important point. So, when this microbe is broken down into smaller pieces, then these pieces are loaded up on a protein called MHC2, Major Histocompatibility Complex 2, class 2. I have actually skipped the whole process of the fragment loading on the MHC2 and then getting out on the surface. We would do that separately. But here, in the overview, all we need to say is that these tiny pieces will be then loaded on MHC2, and then this MHC2 will be extruded or displayed on the surface of the pathogen, surface of the macrophage or neutrophil or the dendritic cell. These cells are called professional antigen presenting cells. Now, here is another delicate definition. Just like antigen is a very specific fragment that can bind with B cells and T cell receptors, loading an antigen onto a major histocompatibility complex protein and then exhibiting or displaying that antigen outside on MHC2 or MHC1 is called antigen presentation. So whenever in the immune system discussion you ever come across the words antigen presentation, this is not simple English writing. This is actually a term or a collection of terms that means to display the antigen on the surface of a presenting cell via MHC2 or MHC1. So now we know what is phagocytosis, eating by the cells. We know the cells that can do phagocytosis in the innate arm, macrophage, dendritic cells, neutrophils. We know the end result of phagocytosis, that is breakdown of the pathogen or microbe. We know that macrophage and dendritic cells can present that on MHC2, and that is going to be called antigen presentation. We discussed yesterday, neutrophils do not do antigen presentation. They just break down the pathogen to eliminate it, but they do not do a presentation. So, one more point about the antigen presentation. This is such an important point. This, if I could say, where is the core concept of the whole immunology, then that concept is this following one. The concept is when the antigen is presented, who is it presented to? It is presented to T helper cells, which are part of the adaptive arm of the immune system. This little process of antigen presentation activates the adaptive arm of the immune system. Without this, we will actually not be able to live for a longer time. This one concept is the basis core function of the immune system. This is how innate arm will trigger or activate the adaptive arm, and the whole immune system will become engaged in helping clear the pathogen. And why is this so important? Look, between yesterday and today, we are seeing that innate arm has cells, it has proteins. Cells are very powerful. They react very fast as well. They can pick up the pathogens, they can destroy the pathogens, they can then release inflammatory markers and cause inflammation as well. They can cause debris removal, they can do inflammation, repair, healing. They can participate in those functions. So it seems like natural immunity or innate arm is sufficient. But sometimes babies are born with a disease called severe combined immunodeficiency syndrome or disease, SCID. In that disease, the baby has no adaptive arm at all, or not functional adaptive arm. Their T cells and B cells are not correctly produced or not correctly functioning. Such babies actually have an intact innate immune arm. Even then, these babies get recurrent, repeated infections and life-threatening infections. So if the innate arm, with all of its power, was sufficient, then we would have been okay in SCID. This proves that adaptive arm's contribution is very important. And this little concept, this little mechanism of antigen presentation is what loops in the adaptive arm. If this one area is broken down, or if a pathogen learns to evade this one process, then our immune system cannot become fully functional. You would learn in the later discussions that adaptive arm then returns the favor and amplifies the innate arm by releasing gamma interferon and many other cytokines and chemokines. That is called interleukin 12 gamma interferon axis, or, in my opinion, innate arm, adaptive arm axis to amplify and work together. So, core concept of immunology done. What is that concept? Antigen presentation. Why is it important? Because it activates the adaptive arm. Now we're done with these main concepts. Now we're going to look at the other cells. So, innate arm, still, effector cells, neutrophils. We'll actually go in detail for the neutrophils later on. This is a high-level discussion. Neutrophil, if you see here, under the microscope, they look neutral in color. They look light pink in their color. Why do we say neutral in their color? Because they don't really pick up the dye with which they are being stained. So they should have either become red for the acidic dye or acidic proteins binding with the basic dye, or they should have become blue. That means negative proteins binding with positive dye. Instead, these cells do not really bind much. So they appear neutral in color, transparent in color under the microscope. Neutrophils are phagocytic, so just like macrophage and dendritic cells. However, they do not perform antigen presentation. They're a little selfish. They don't do it. They're lazy, but they're very, very active as well. They perform respiratory bursts. They break down the pathogen in a different way compared to the macrophage and dendritic cells. Neutrophils actually burn the pathogen by using oxygen bursts. Their granules are lysosomal and pink in color, light pink. These neutrophils can also release toxic enzymes that can actually cause damage to our tissue. That is a problem with the neutrophils. Actually, it's a problem with all the immune system, but especially the neutrophils. Neutrophils will pick up a pathogen microbe, break it down. In that process, they would also release so many chemical substances that those would cause tissue damage. And then, if somebody has neutropenia, that is less number of neutrophils, then that person develops a lot of bacterial and fungal infections, which tells us that neutrophils play a role in helping against bacteria and fungi as well, in addition to the viruses. But especially they are good for bacteria and fungi. Next, Paul Wolf says, what is a respiratory burst? So we'll discuss that in detail. A respiratory burst is where the neutrophils would form reactive oxygen species and then use them like little fireballs and attack the membranes and the tissues of the microbe. They don't have tissues. The smaller molecules of the microbe, neutrophils would throw reactive oxygen species on them, radical oxygen species on these pathogens, and the pathogens would just break down. We call it they are burnt up. That is called respiratory burst. Because it uses oxygen, neutrophils need oxygen to do their function. This is why we say they respire and they create a burst. That burst breaks down the pathogen. Okay, so next one, eosinophils. Eosinophils stain red with the right stain. Right stain is actually the name, WRIGHT, right? Now, as much as we know, and it is generally said that eosinophils help against protozoas or the worms, it's actually not fully known that that is exactly the function they are supposed to perform. Eosinophils' actual protective function is still not clearly elucidated. We still do not actually know it. However, we know that their count is increased in certain worm infections. For example, in nematodes or other worms of similar species. Their count is also increased in hypersensitivity reactions. So maybe they have a function there. At the same time, neutrophils have an enzyme in them which is called histaminases or histaminase. Histaminase, of course, as the name says, whenever, excuse me, whenever we have the A's postfix with a word, that simply means breakdown or an enzyme. Histaminase would be an enzyme that breaks down histamine. Now we know that histamine is a part of inflammatory system. So histaminase should, in theory, neutralize the histamine and reduce inflammation. So as much as we see that is eosinophilia, meaning increased number of eosinophils, occur in hypersensitivity reactions, at the same time, neutrophils actually have histaminases. So maybe their count is increased to help release histaminases more to neutralize the histamine being produced in the hypersensitivity reactions. Or maybe eosinophils actually participate in creating and maintaining allergic reactions. Their main protein that stains red is called the major basic protein. This protein is positively charged and it picks up the negative part of the dye and then it looks red. And it is the major basic protein. So if you see here, I made these little eosinophils biting the worm. So they really are not biting the worm. What they do is they release the major basic protein. Major basic protein then damages the worm's skin and causes the wounds and injuries to occur and 1,000 cuts and the worm dies. These cells are actually microscopic. Worms are macroscopic. Even then, these cells gather around the worm, release major basic protein, and destroy it. Then is the basophils and mast cells. Just like monocytes and macrophages. What do you think is the difference between monocyte and macrophage? Monocyte stays in the circulation and can get into the tissue, where once it gets in the tissue, a monocyte will become a macrophage or a dendritic cells. Although there is a generation of monocytes that can live at the boundaries of the body tissues and stay as monocytes, but they tend to convert to macrophages mostly and to dendritic cells and live in the tissues that way. Similarly, basophils, we call them basophil because they're basophil. Phil means liking. These cells, they have negative charged particles in them that like the positive dye. So they're basic in color, blue in color, or they pick up blue color. Basophils live in the circulation, just like monocytes live in the circulation. When basophils leave the vascular system, the circulation, and enter the tissue, they can become residents there, just like monocytes can become residents. And when they become residents or become fixed in the tissue, then the basophils are called mast cells. When monocytes become residents, they are macrophages or dendritic cells. When basophils become residents, they are mast cells. That means majority of the properties of these two cells are the same, except their location. So basophils, blue dying cells. And if you see here in this little diagram, I have made a lot of antibodies on them. Basophils and mast cells have a huge number of receptors on them to bind with the IgE, immunoglobulin E. We haven't yet studied IgE in this session. So we'll see that in the future as we do the B cells and immunoglobulins or antibodies, that there are various kinds of antibodies and IgE is one of them. Now, basophils and mast cells are part of the innate arm. However, for them to be fully functional, they need the adaptive arm, B cells, to produce antibodies, which will then lodge on their surface. These antibodies will decorate their surface. Once these basophils or mast cells are decorated with IgE, now they are fully functional and primed, as we call them. They're ready to go. If you expose these primed basophils or mast cells to whatever allergen they bind with, or whatever antigen they bind with, we normally call them allergens, then these cells would degranulate. They would release their toxic molecules and cause a lot of damage. They can cause hypersensitivity reactions, the simplest of which is the urticaria or hives, and the severe form is anaphylactic shock, even killing a person. So beautiful cells, but really, really problematic as well. So they circulate in the blood, basophils. Mast cells are fixed. Both cells have IgE receptors. IgE in turn acts as a receptor. Once IgE is decorated on them, then the IgE becomes a receptor. Cross-linking of two closely present IgE would cause the activation of the cell, and the cell would degranulate. These cells have histamines, proteolytic enzyme. Proteolytic enzyme mean enzyme that breaks down the proteins. Heparin, prostaglandins, leukotrienes, all really dangerous. They're good as well, but dangerous molecules. They cause inflammation, severe inflammation, or allergic hypersensitivity reaction, we call them. And we would discuss what these reactions are. When we talk about the basophils and mast cells, the definitions change a little bit. The antigen that binds with them is usually called allergen, and we'll discuss why it is called allergen later on. The response of these cells and the inflammation as a result is usually called hypersensitivity reactions instead of just simply inflammation. But underlying mechanisms are similar. Mast cells, as I said before, the only difference from basophil, this cell lives in the tissues, especially under the skin. That is why hives occur under the skin, because of the inflammation under the skin. Under the mucous membranes, that is why respiratory membrane and when somebody is getting hypersensitivity reaction, their lips start swelling, because the respiratory mucous membranes would start swelling up and the person would start choking. So the mast cells are abundantly present under the skin and mucous membranes. So when they degranulate, when they become upset, of course, the simplest thing they would do is urticarias and hives, itching, burning skin. And the worst is shock, anaphylactic shock, even causing death. And the final cell, the final effector cells. So effector cells mean the cells that do some function of fighting with the pathogens. So this effector cell is the natural killer cell. Natural killer cell is very interesting that it seems like it comes from the adaptive arm cells or that line. However, natural killer cells do not have memory like adaptive arm B or T cells have. Natural killer cells are not specific like B or T cells of the adaptive arm are. These are specific cells. And natural killer cells also work with the patterns instead of the antigens. So they probably come from the adaptive arm, but they have joined the ranks on the innate side, and they have developed the capabilities of working as an innate cell. These are my favorite cells, NK cells. I always make them with this little eye patch on them and with lots of hands. They are favorite because of their function, but I also call them perverts. Why? Because natural killer cell, if you can see so many hands on them, natural killer cells have a ton of receptors on them. And when these cells are passing by any other cell, they will touch it. And on a regular normal cell, they will count, the natural killer cells. Natural killer cells will count or assess the density of MHC1 molecular clusters. And we'll discuss MHC1 later on as well. That is major histocompatibility complex number 1, which is also for antigen presentation by regular cells. So the MHC1 density becomes less on a normal regular cell that is sick. For example, if it has some virus sitting in it, or a fungus or a bacterium, intracellular microbes. The microbes are generally weak. From a location point of view, you can say they are extracellular and intracellular. Intracellulars are those that have to be in the cell. All viruses have to be in the cell. Some bacteria like to be in the cell as well. Some fungi like to be inside the cell too. Intracellular microbes, when they hijack the cell's machinery and start using it for their own purpose, the result is that the cell's machinery and cell's proteins are not formed. And one of the indication of that is the reduction in surface proteins. So all surface proteins are reduced. However, natural killer cells have decided to look for MHC1 density. On the other hand, so this is on the regular cells. On the other hand, the cancer cells display multiple abnormal proteins on their surfaces. Couple of such proteins are called MICA, mica, or MICB. These stand for Major Histocompatibility Complex Class 1 Related Chain Protein A or B. So here, MIC1 chain related protein A or B. Kind of like MSC, but different. Natural killer cells also can sense the presence of these proteins. Here on the regular cell, they are sensing the absence of the proteins. On the cancer cells, they are looking for the presence of the mica proteins or MICB proteins. In either way, if on a regular cell, MIC1 is less, or if on a cancer cell, mica is present, then the natural killer cell is a natural killer. It is going to kill them right on the spot. It's not going to wait for anyone. It's going to kill them right there. And how does it do that? I always make these cytotoxic T cell as well to have these granzymes here and perforins. So what these cells do, the cytotoxic cells, they make a hole in the target cell, and through that they throw enzymes inside the cell that would digest and destroy that cell. So here, the hole making protein is called perforin. The protein that would activate the cell destruction is called granzyme. When the granzyme is thrown inside a cell, it will cause the triggering of the apoptotic functions of the cell. Apoptotic function means the function of a cell to cause or to trigger the cell death. You can actually order a cell to die. And when the cell would die, that is called apoptosis. NK cells and cytotoxic T cells can trigger cellular apoptosis. So this is the discussion. I am still very, very excited. I become so excited I cannot even speak sometimes correctly, but this is the second lecture, immune system function. Tomorrow we'll do a COVID lecture. However, I'm preparing more for the immunology as well. So with this, please do me a favor. There are links in the description. You can support this work. The easiest is like, subscribe, and share. But if you would like to support this work further, there are links in the description. You can become a patron. You can use PayPal. You can buy me a coffee, and more things are there. Cool. So with this, I would see you tomorrow. Have a good day. So John says we're getting excited too. I am too excited about it. It is the second lecture, but I have done a ton of immunology lectures before. Now we are cruising towards a book, and I would really, really hope that I'm able to present these and then write a book. So your support, so of course, folks are supporting financially, but your support of liking, subscribing, sharing also helps a lot. So with this, I would see you tomorrow. Bye-bye for now.